Hello friends, and welcome back to the channel. As usual, it's beautiful to have you here, and we'll continue in our discourse. Today we are looking at we've been looking at forensic microbiology, which is the aspect of microbiology that works on determining if an agent, a biological agent, or its products have been used to commit a crime. And if that has happened, as a scientist, what are your interventions? What do you do, and how do you help the authorities, the police, the government? How do you help them to be able to fish out what particular organism was used, and then the origin of the organism, and by extension, who are the people who have been perpetrating this crime? Now we're getting down to a lot more technical areas here where we discuss basically the techniques that are used in forensic microbiology can be categorized into two broad groups. We have the culture-based methods and the molecular methods. Now, the culture-based methods are old methods that have been used in microbiology, but are still in use today. And when you talk about the culture-based methods, you're talking about methods where we use different kinds of culture media, selective media, differential media, all these different kinds of media. And there are so many: nutrient agar, MRS agar, a lot, McConkey, very many, Muller, Hilton, and so many different types of agars that are used. And when we're talking about cultural media, you have two broad classes, two types. You have the ones that are in the solid form are the ones we refer to as the agar, and the ones that are in the liquid form are the ones we refer to as the broth. So many of the cultural media can also be in liquid form. So you can have nutrient broth, you can have every different kinds of broths. So the idea being that we are saying that when you're trying to identify an organism that can, or its product which has been used as a bioweapon, you have one of two routes to go. You can either go through the culture method route, or you do molecular analysis. Now, if we're doing culture method, we're saying that we are growing these organisms on culture media. This media could either be in solid form or in the liquid form, and these are important for us to know. Now, the culture-based methods are essential. They're essential because they help us to be able to isolate and identify organisms. So different samples can be taken from, wherever our interest is. Can be taken from the person. If a person who has been a victim of such a crime, samples can be taken from the mouth, from the skin, from the nose, from different aspects of the individual's body. You can take sample and then try to culture from those samples, try to culture out different types of microorganisms. And this has been done, and this is being done on a daily basis. Now, there are advantages to doing a culture. Now, the advantages to doing a culture include: one, it helps us in classic identification. Culture methods are the cornerstone of microbiology. So even in very advanced laboratories, we still do culturing because it's the cornerstone of microbiology. And so you have they help us to do classic identification. So different types of microorganisms, we know their features, we know their characteristics, we know how they respond to different biochemical tests. So all of these are done to use the classic method to try to identify our microorganisms. And this is quite important. I talked before about a word called morphology. So you want to look at shape, size, different features that these microorganisms have, different enzymes which they may carry, or which they carry, which helps them to react in different ways to different types of biochemical tests and assays. This is really important in the culture-based method. So it helps us in classic identification. Also, it helps us with antibiotic susceptibility testing. Now, for an example, somebody is a victim of some kind of bioweapon attack. If it is possible, and if the bioweapon is bacteria, for an example, it can then be possible to make use of antibiotics to use to cure the person of whatever infection or illness that this bacteria has caused. Now, when we culture the microorganisms, it is easy for us to do an antibiotic susceptibility test, which is simply a test, an assay for which we test the efficacy of different antibiotics against this agent of interest. So we culture the microorganism, the bacteria, if it's a bacteria, we culture it on culture medium, and then we subject it to different types of antibiotics and see which antibiotics is effective against that agent. Now, it's a very beautiful test because it helps us to be able to determine what kind of antibiotics that can be prescribed by the doctors and other prescribing agencies for the individual, so that that can be used to fight against that kind of infection. This is possible when we do a culture test. Also, the culture test is good because it helps us to isolate the organism. It helps us to see how viable the organism is in different conditions. It helps us to see what can be used, for prevention. Different analysis can be done on an organism if that organism is culturable. So these are the advantages of being able to culture an organism. But it also has its disadvantage. The disadvantage one is there's time constraints. It's time consuming. So it takes a lot of time to be able to culture the organism. It could take 3 days. It could take 5 days before you have a fully viable organism from your sample. So precious time could be lost while we are struggling trying to culture this organism. That's a disadvantage. Another disadvantage is that it's not every organism that is culturable. Some organisms we have not been able to, find out the conditions and the media and everything that is needed to be able to culture those organisms in the lab. So for such organisms, trying to say that you are going to culture such organisms in the lab is going to prove abortive. It's going to prove an impossible mission. So that is also a disadvantage of culturing. Now, when we leave culturing, we can now go to molecular methods. Remember, I said that when it comes to identification of bioagents, that we have two broad groups. You can either go through the culture route, where we do culturing and isolation in the laboratory, or we can use molecular identification. Now, when we come to molecular identification, there are about 4 different techniques, which we'll just talk about very briefly. There are many techniques for molecular identification, very many techniques. But we'll talk about PCR, which is polymerase chain reaction. We'll talk about metagenomics. We'll talk about whole genome sequencing, and we'll talk about one more. So polymerase chain reaction, we've discussed that in a prior video, where we said that what we do when we do PCR, polymerase chain reaction, is to amplify DNA, to have many different types, or no, not many different copies of the same DNA, not different types now, many different copies of the same DNA. And when you have these many different copies, you have what we call a primer, which we said is like a tag. So you use it to tag these different DNAs, the idea being to just identify the organism. Now, you can also identify for the presence of other things in the organism. You may be targeting some particular kind of virulence genes, some genes that make the organism to be pathogenic in nature. You could be targeting those genes. You could be targeting other antimicrobial genes. So whatever it is that you're targeting, you can use your PCR and use your primers to tag DNA that has been amplified, that has been made many copies of that DNA, to identify for the presence of either the organism or genes that you might be interested in. This is what we do in PCR, and it is a very important technique that has been used in forensic microbiology.